There's a fungus out there that doesn't just infect insects, it operates on timing. Right around sunset, it releases chemical signals that cause the host to climb higher before it dies. Scientists took fluid from an infected fly, introduced it into a healthy one, and the second fly began climbing as well. No visible fungal growth inside the new host, just the chemistry. So, what exactly is in that signal, and how does a fungus synchronize its activity with time so precisely? The fungus is called Entomology muscae, and it has been operating this way for millions of years. Long before we understood parasitism or chemical communication, this organism had already adapted to influence behavior through internal signaling. Because the key detail here isn't physical control. The fungus doesn't rebuild the fly's brain or form direct connections with its nervous system. It interacts through the body's existing processes, sending signals that the system responds to. That signal appears to come in the form of compounds that resemble the fly's own internal chemistry. The body processes it as a familiar instruction. Move upward, continue climbing, don't stop. And it responds because it can't distinguish between internal regulation and external input. From the system's perspective, it's following its own signals, even though those signals are being influenced from within. This is where the pattern becomes more precise. The timing isn't random. Sunset aligns with when other flies are more likely to be resting on elevated surfaces, which increases the likelihood that spores released from a high position will spread efficiently. The behavior isn't just a side effect of infection. It reflects a timing-dependent process that improves transmission. The system operates through chemistry and environmental cues, aligning internal signals with external conditions. And if a set of compounds can carry that level of instruction, the question extends beyond this single case. It raises how many other interactions in nature rely on similar chemical signaling, where organisms respond to inputs that resemble their own internal processes. The distinction between internal regulation and external influence becomes less clear when both use the same language. From a systems perspective, the objective is straightforward. The organism depends on a host that remains mobile long enough to distribute spores effectively, so maintaining function is essential even as internal resources are being used. That balance requires precise timing and regulation, allowing the host to continue operating while the underlying process progresses. The infection starts on the outside. Spores land on the insect's cuticle, the hard outer shell, and the fungus releases enzymes like chitinases that allow it to pass through that barrier. Once inside, fungal cells called hyphal bodies enter the hemolymph, the insect's internal fluid, and from there, they circulate throughout the body, reaching multiple tissues as part of the normal flow. What happens next is where the process becomes more selective. The organism doesn't consume tissue at random. It draws first from fat reserves and less critical resources, using enzymes such as trehalase to break down trehalose, a primary energy source in the insect's system. This allows it to support its own growth while leaving key structures like muscles and the nervous system largely functional. Movement continues. Climbing behavior remains intact. The host stays active for a period, even as internal conditions change. It's often assumed that this kind of behavioral shift must come from direct interaction with the brain, as if the system is being physically altered at its control center. But research from Penn State University tested that idea using fluorescent microscopy to track where fungal cells actually appear. What they found was that fungal presence inside the brain itself is minimal. The structure remains largely unaffected, even while observable behavior changes significantly. So if the brain isn't being directly entered, what's actually happening? The fungal growth concentrates around the outside of the brain and extends through muscle tissue, especially in the legs and jaw. Instead of altering signals at their source, it interacts with how those signals are carried and expressed through the body. The communication between the brain and muscles still exists, but it's being influenced along the pathways that connect them. In ants infected with Ophiocordyceps, researchers observed the fungus growing into muscle fibers, particularly in the mandibles, the jaw structures. This growth affects how those muscles contract, leading to sustained gripping behavior. The ant attaches to a surface and remains fixed there. Rather than originating as a direct decision from the brain, the behavior emerges from changes in how muscle activity is regulated at the tissue level. What this suggests is that the behavioral shift operates through the body's existing circulation and peripheral systems, not through direct interaction with the central nervous structure. Compounds move through the hemolymph, reaching muscles and surrounding tissues, where they influence activity patterns. The effect is distributed, occurring across multiple parts of the system, rather than at a single control point. From a systems perspective, this represents a different kind of interaction than most people expect. The organism maintains host function by selectively using available resources while preserving key structures needed for movement and positioning. Energy reserves are reduced first, while muscles and critical mechanical functions remain active longer. The process reflects a balance between resource use and functional maintenance, supporting the organism's life cycle without requiring direct alteration of the central control system. But the process operates through chemical signaling distributed across the body, moving through fluid rather than being tied to a single structure, which raises a more complex question. A fungus doesn't have a nervous system of its own, no centralized organ for processing time, so how does its activity align so precisely with a specific moment in the day? Infected flies don't show this behavior randomly. It occurs within a narrow window near sunset, and that level of consistency points to an underlying timing mechanism. Researchers studying Entomophthora muscae infections in Drosophila melanogaster documented this pattern in detail. The flies move to elevated positions near sunset, attach to a surface, raise their wings, and then remain there. The timing repeats with remarkable regularity across individuals. That consistency suggests the process is linked to something the fly's own system is already tracking. Like many organisms, the fly maintains an internal cycle that follows the roughly 24-hour light-dark pattern and coordinates behavior across the day. Within that system, specific neurons, known as DN1PS cells, contribute to regulating activity and movement in relation to time. When researchers altered the function of those DN1P neurons in infected flies, the climbing behavior decreased significantly. This indicates that the timing isn't generated independently, but is aligned with the host's existing biological rhythm. The organism's internal clock provides the structure, and the chemical signals operate within that framework to produce the observed behavior. The hemolymph transfer experiment is where the mechanism becomes clearer. Researchers took fluid from a fly that was already infected and showing climbing behavior, then introduced it into a healthy fly with no fungal presence. The healthy fly began climbing as well. No visible infection, no fungal growth inside its body, just the chemical components carried in that fluid producing the same behavioral pattern. That result shows the signaling isn't confined to fungal cells. It's soluble, it moves through circulation, and it can influence a system even in the absence of the organism that produced it. What's present in that hemolymph is still being studied, but genomic analysis of Entomopthora muscae has identified some relevant candidates. The genome includes genes similar to white-collar 1, a light-responsive gene found in other fungi such as Neurospora crassa, where it plays a role in detecting day-night cycles. In those systems, the gene helps coordinate internal processes with changes in environmental light. If a similar mechanism is active here, it suggests that the organism may align its activity with external light conditions, producing signals in coordination with specific times of day. At the same time, the host organism is already tracking the same light-dark cycle. through its own internal rhythm, including neurons involved in regulating movement and activity. The summiting behavior isn't simply a late-stage effect of infection. It appears as a timed output, shaped over long evolutionary periods and aligned with the host's own circadian rhythm and the release of compounds into circulation. An organism without a nervous system interacts with one that already exists, relying on its function rather than replacing it, and the process only needs to occur at a specific moment to be effective. What this research shows is that changes in behavior don't necessarily originate from a central processing system. They can emerge from chemistry, timing, and interaction with an existing biological framework. The organism doesn't process information in the way a brain does. Instead, it operates through established biochemical patterns, with the host's nervous system continuing to carry out the resulting activity. Scientists studying these interactions see broader implications for how microorganisms influence biological systems more generally, including within the human gut. Many of these processes rely on chemical signaling that is only partially understood, and the tools needed to fully map these interactions are still being developed. As of recent research, at least 35 species within the Ophiocordyceps group have been documented to affect ant behavior, with many more likely remaining unobserved. The full extent of these systems is still being uncovered,